animals interact with their conspecific. In this case, large rat, 300 grams, interact with smaller rat, like 100, 150 grams. And spend some time to know and so on. And this interaction is inhibited when compared to the non-animal object in the stressed animals. The big rat, when stressed, when kept in these immobilized conditions, does not want to be so social anymore. So the stress impairs social function. Okay, which kind of a mental function. And this impairment is prevented by MMP9 inhibitor infused into the hippocampus. Okay? These were animals. Now coming more to humans. I mentioned this issue about binding neurexin neuroligin. And neuroligin being cleaved by MMP9, neuroligin 1 by MMP9 inactivated neurons that activated synapses. And I'm taking from Thomas Sidov review some years ago. Neurexin and neuroligins have been recently implicated in autism and other cognitive diseases. Actually, when you look for the literature of gene association, polymorphism, combinable variants, this kind of stuff, you find that neuroligins and neurexins are more often associated with autism and schizophrenia and some other disorders. FMRP is fragile X mental retardation protein. This is the protein that is deficient in the disease called Fragile X syndrome in humans. And this is most common mental disability in boys, is X-linked chromosomes. So mutation in this gene produces intellectual disability, autistic spectrum disorder symptoms. Actually, the recent, yesterday or 2 days ago, paper, two papers in Nature that again link the most often genetic variation in autism spectrum disorders with FMRP. Somehow it's interactive. Epilepsy, 25%. Neurons exhibit numerous long philopodia with prolonged structures. And obviously, since the protein is missing in humans, when we make the knockout in animals, it actually very well reproduces. It's one of the best models to study this disorder. Irina et al. In California, in Riverside, found that when she looked at the culture of this FMRP knockout mice, again there are longer philopodia, but this could be returned to normal, more mushroom shaped with minocycline. And minocycline, among the other functions, is an inhibitor of MMP9, especially MMP9. So there were trials in the US looking whether minocycline could be beneficial in this disorder, Fragile X syndrome. And the colleagues from Sacramento who asked us to collaborate really reported that this minocycline given for 3 months in children were beneficial. And they asked us to look for MMP9 levels in the blood, in the serum of these patients, and during the treatment, again, MMP9 was decreased, MMP2 was not affected. After the treatment, the MMP9 came back to the levels. Again, now, mental function in humans. Addiction are disorders that are based initially on the plasticity. Later on, there could be neurodegeneration and so on, but initially, there's increased plasticity that leads to addiction. Since MMP9 is linked to plasticity, we asked our colleagues from Szczecin, Jerzy Samochowiec, who is a chief psychiatrist at the Medical University of Szczecin, and he studies alcoholics, for the study on MMP9 gene polymorphism. There is a polymorphism far upstream of transcription initiation site, minus 1562, that affects MMP9 gene expression. 25% of us have higher level of expression, 75% lower level of expression. Yes? In whatever population. It was also studied in other population, different countries. Interestingly, in the family of alcoholics, it occurs twice more often, more frequent, this polymorphism. This actually shows that then we were interested whether what is the effect of MMP9 on alcoholism, and together with Kasia Radwańska, actually, she mostly developed a special system to study alcoholism in mice in these intelligent ages, and one of the things that can measure is motivation, which could be measured by how many nose pokes you have to do in order to get to the liquid, like alcohol liquid there. If you need more nose pokes, more repeated nose pokes to get alcohol, you are more motivated to get it. And MMP9 knockouts seem to be deficient in this motivation phase. So it's going big. So we got animal data that MMP9 is involved in physiology, plasticity, memory. Went to the human mind of the alcoholics, found that there is something going on. Now we go back to mice to explain the mechanism of this. Vizela dog had various mental disorders, and post-mortem is not very well diagnosed yet, but is suspected to have epilepsy, addiction, schizophrenia, bipolar disorder. So it shows the complexity also of definition of those disorders. So I also asked Janusz Rybakowski, who is chief psychiatrist in Poznań Medical University, and he studies bipolar patients' schizophrenia to look for this polymorphism, and he found some association in schizophrenia and bipolar. There are differences. And then Hannelore and I, our friends from Max Planck Institute for Experimental Medicine in Göttingen, they developed a database over 1,000 patients with schizophrenia all over Germany and the appropriate number of controls. That each of these patients, called GRASS, Göttingen Research Association of Schizophrenia, and each of these patients provided like 3,000 data points, including blood samples, psychiatric scores, psychological scores, whatever. And I asked her to interrogate this database, those patients, with MMP9 polymorphism. And she came out with another polymorphism she discovered, which is called RS2544. And she found that this polymorphism in males and females, in humans, correlates with positive symptoms of schizophrenia, such as delusion and hallucinations, okay? And a bit differentiating between females and males, okay? We looked at this polymorphism, what it does, with help of colleagues from Poznań bioinformaticians. And the polymorphism is located here, 3' UTR nucleotide number 3, and by itself does not seem to affect anything important. But when looking at the secondary structure of RNA, it modifies also its partner here, and this 109, 110, whatever, 109 nucleotides that binds to this module, depends on the polymorphism. Either you see this, there are 4 or 5 base pairs here. This one, secondary, is located with the G quarter that I mentioned that binds FMRP. Okay? So we follow this, and it looks that since FMRP affects the level of MMP9, that this level depends on the polymorphism variant, produces different level of expression. So, to conclude, first, I wanted to provide you with the message that C4's MMP9 link is important for mental capacities, starting from memory as a window to understand this, or reveal something to the mind even of the humans and diseased mind of the humans. But I don't want to give the impression that MMP9 is the only or the most important key to understand those functions. Okay? In biology, as you know, and hopefully you are listening to Professor Jig here on this hall, some of you, and I'm sure that he should mention this, we have this holism versus reductionism. Biology is about holism. You cannot explain the structure, not understanding components and emergent properties of the whole structure, coming from evolution, interaction, ecology, whatever. So what I like to say is that these stones that build this beautiful cathedral in Strasbourg do not explain all this craftsman work. Okay? They are just components, building blocks. MMP9 is one of the first building blocks that is so well characterized. But this requires a lot of effort to understand what is this human work that produces this beautiful thing here. So finally, I would like to thank people who are involved in the work, either at the Nencki Institute and medical apparatus or other agencies that support our work over those years. And also, finally, an addendum. I was asked by Peter Somogy from Oxford to advertise among students here that Peter created a foundation called Stephen Kufler Foundation. Stephen Kufler was a very famous scientist, originating from our part of Europe, and a great mentor of Wiesel, Kandel, Hubel, Nobel laureate. So to commemorate Kufler, Peter created a foundation. That major thing is to award scholarship to Central European students. So you will have all the information on the website if you would like to apply for this award. Thank you very much for your attention.